Welcome to today's Explainer. We're diving straight into one of the most highly regulated, just astonishingly precise biological factories in the known universe, your very own cells. We are going to be exploring the cell cycle, which is that microscopic sequence of events that cells use to grow and divide every single day. It all starts with this massive, macro-level question. How do our bodies constantly rebuild themselves? I mean, really think about it. Throughout your entire lifespan, from early development all the way through adulthood, your tissues are constantly growing, they're maintaining themselves, and they're repairing damage. And it isn't magic. It's the direct result of microscopic cellular machinery working completely around the clock. Okay, let's dive into this. Our itinerary today covers 5 main stops. First, why cells must divide. Then, the long prep, followed by the quick split. After that, we'll look at exiting the cycle, and finally, the cost of losing control. So, part 1, why cells must divide, or rebuilding from the inside out. Let's anchor this whole biology concept in a super relatable, highly physical example right inside your own body. Take the human gut. The epithelial cells lining your intestinal tract deal with an incredibly harsh environment every single day. Because of this, they have a super rapid turnover rate. They literally have to be replaced continuously just to maintain the structure and function of the tissue. And the absolute workhorses behind this constant regeneration are epithelial stem cells, which just sort of hang out in these little pockets called the crypts of the intestinal lining. This actually perfectly illustrates a beautiful division of labor in our tissues. On one hand, you've got stem cells, and when they divide, they either make more stem cells or they create daughter cells that will eventually mature. But then, contrast that with the mature epithelial cells. These guys have permanently exited the cell cycle. No more dividing for them. They migrate out of the crypts, do their digestive jobs, and are ultimately shed right into the digestive tract. It's basically this continuous, never-ending conveyor belt of life and death. Moving on to part 2, the long prep, representing 23 hours of meticulous preparation. Now, the sheer amount of time a cell spends just carefully growing and preparing for division is kind of mind-blowing. For an average cultured human cell on a 24-hour cycle, a staggering 23 hours is spent in what's called interphase, just getting ready. Only a tiny 1-hour sliver is actually dedicated to the physical division itself, the M phase. During this incredibly long prep time, the cell is doing its normal bodily functions, sure, but it's also significantly growing in size and meticulously replicating its DNA and organelles. So breaking down those 23 hours, interphase actually runs in 3 distinct stages. The longest is gap 1 or G1, which takes about 11 hours just to grow and produce the proteins necessary for replication. Then comes the S phase, or synthesis. This takes 8 hours to synthesize and completely replicate the DNA, which is packaged up as chromatin. And finally, gap 2, or G2, takes another 3 to 4 hours for a final spurt of growth before the cell is truly, finally ready to go. But before the cell can just move from the G1 growth phase into the S phase, it hits a major roadblock. You can think of this as a very strict factory checkpoint inspector. The G1/S checkpoint sits right at the end of that 11-hour window. Its sole critical job is to make absolutely sure the cell's DNA is entirely intact and completely undamaged before any replication even begins. It's there to completely prevent catastrophic errors from being copied over. Once it passes that strict inspection, we enter the S phase, where the magic number is 46. During this massive 8-hour window, every single one of our 46 chromosomes, half from mom, half from dad, must be perfectly duplicated. This intense replication process creates identical sister chromatids for every single chromosome, and they're held tightly together by a protein complex that's very appropriately named cohesin. So we now have 2 identical copies of each maternal chromosome and 2 identical copies of each paternal chromosome. This level of precision is absolutely crucial. It ensures that when the parent cell eventually does divide, there is literally 0 loss and 0 gain of genetic material. All right, so after the S phase comes the G2 growth phase. And at the very end of that, we meet our second factory inspector, the G2/M checkpoint. This gatekeeper is there to guarantee that the DNA is fully replicated and that any damage incurred during the synthesis phase is completely repaired. Only then can the cell finally commit to the actual physical split. Which brings us to part 3, the quick split, 1 hour of dramatic biological action. We are officially leaving the slow, careful preparation behind and entering a rapid, super intense 1 hour window of pure, dramatic biological action. Here is exactly what goes down in that frantic hour. The chromatin condenses super tightly into that classic chromosomal X shape we've all seen in textbooks. They line up right down the middle, attached to a network of proteins called the mitotic spindle, and then the spindle's microtubules shorten. They physically wrench those identical sister chromatids to opposite sides of the cell before entirely new nuclear membranes can form. But even amidst all this intense cellular chaos, biology really doesn't leave anything to chance. We actually have a third inspector here, the spindle assembly checkpoint. Right before those chromatids are ripped apart, this checkpoint hits pause on the whole factory to ensure every single chromosome is perfectly aligned and attached to the spindle. It guarantees a perfectly even distribution. No chromosome left behind, basically. Finally, the whole cell undergoes cytokinesis, which is the physical dividing of the cytoplasm and plasma membrane. The glorious outcome of the M phase is two genetically identical diploid daughter cells. One parent yields two perfect genetic clones. Though, it is worth noting that the other components, like the organelles and the cytosol, are just sort of randomly divvied up in the split. It's really the DNA that gets the VIP precision treatment. Part 4. Exiting the cycle. Where do cells actually go when they stop dividing? So the division is over. What happens when a newly minted daughter cell faces its future? Well, a cell re-enters G1, but from there, it actually has several totally distinct fates. Some, like our gut stem cells we talked about earlier, they just keep cycling forever. Most cells, however, exit into a non-cycling phase called G0. Sometimes this is temporary, like liver cells wait here, but they can jump back into the cycle in response to organ damage, which is what gives the liver its amazing regenerative power. Other cells, like our mature gut cells, they enter a permanent G0. But here's where it gets wild. If exposed to overwhelming stress or DNA damage, a cell might be forced to undergo apoptosis, which is programmed cell death. Basically, it self-destructs for the greater good. Or, under mild stress, it might enter senescence, which is a non-dividing, pro-inflammatory state. Which leads us to part 5, the cost of losing control, or what happens when the biological factory breaks down. Everything we've discussed so far relies on just absolute, unbelievable precision. But what happens when the factory breaks down? Remember that senescent state I just mentioned? When cells experience mild stress and exit the cycle into senescence, they don't just disappear. They hang around, they accumulate. And this buildup of senescent cells throughout the body is directly associated with tissue damage, the onset of various diseases, and ultimately, the overall macro process of human aging itself. But for sure, the absolute most severe consequence happens if those incredibly strict factory checkpoints fail. As multicellular organisms, we depend entirely on accurate, tightly controlled cell division. If those signaling mechanisms dysregulate, errors in replication just pass right on through, and that leads to uncontrolled cell division. The exact same cycle that allows our tissues to heal and grow becomes the devastating engine of excessive growth and cancer. Which leaves us with this final, really profound thought to consider. If our very survival, our growth, and our daily healing depend entirely on this razor's edge of cellular control, could mastering these microscopic checkpoints hold the ultimate key to stopping aging and curing cancer altogether?